Many different substances in the cells. We're moving on. Any questions more to that process, or we're pretty okay with that for now? I mean, most of it we need to know these four phases of mitosis. Meiosis, if you look at the test tube, I just wanted to get the fundamental of that, the genetic material amount. Now we need to spend a little more time on substances that need to move in and out of the cell. And some of them move in and out of the cell effortlessly, while others are dragged across the cell membrane. Active parts, active transports that have to be dragged require ATP. Passive transports don't. So when we look at the driving force for the passive processes, very often, or it is called, it's the concentration gradient. That's really the driving force. So when you visualize, you have a membrane, you have a lot of molecules on one side of the membrane and nothing on the other side. If you just open a door and you let these molecules go around, the place, there's a lot of them, and you want to go to the place where there's not many in. Because the place where there's many in is a high concentration. And so if you have a lot and then a membrane and nothing, the gradient of that concentration, how steep that concentration is very heavy. And so that process is passive. Like if you go on a bike from up here down back to Oakland to the freeway, you don't have to do nothing. Well, you need to brake because it's steep. But if you come the other way, you've got to pedal real hard. So that's the concentration gradient in the cells is the same as the steepness gradient in that example. And so when we look at passive ones, passive processes, we have a multitude of them being described. Can you name some of them? Diffusion, for example. Osmosis. Filtration. Those are pretty much the three main ones. Yes?
Can a passive process generate ATP?
It is actually a passive process that does generate ATP, because in the mitochondria, you separate the hydrogen ions out to one side and make the gradient that's really steep, but then you open a little thing and that coming through attaches the phosphate back to the ATP. And so it is very much the same principles. Yeah, we use these principles as much as we can. So diffusion is the simple thing when you take a sugar cube, you put it in water, and the sugar might disperse to the whole water. That's simple diffusion. We have, for example, an oxygen exchange into the red blood cell. Simply, there is more oxygen in the alveoli in the lung than in the blood that passes through. So the gradient is higher in the alveoli, so the oxygen wants to move right into the red blood cells, wants to move right through there. That's simple diffusion. Facilitated diffusion is when we need a channel. That's the example with the glucose. So the glucose will not go into the cell unless there is the insulin says you go in and the channel is open. But once that channel is open, the glucose passively flows in. Because the cell makes it, as soon as the glucose comes into the cell, it tags it, I think it's a phosphate, and at that point, chemically, it doesn't look like a glucose anymore, so it's like the concentration is still there. The gradient of the concentration is still there. The glucose still wants to go into the cell, and it's outside. So it's kind of interesting. It's a little trickery with that, the way they do it. But that's facilitated diffusion, because if you don't have the channel open, it ain't happening. It's dependent on the channel. Then osmosis, that's the cool one. If you simply put, osmosis is the diffusion of water. So if you have, let's say you have a U-shaped beaker here, and you have a membrane in between here, and this membrane only lets water through. Only water can penetrate this, nothing else, not the solute. If you look at chemical things, you've got solvent and solute, right? Solute is the stuff. So these dots here are solutes. So you see, on one side here, we have many more solutes than on the other side. If we have a membrane that doesn't let nothing through, not even water, it's got to be like this. However, if we make this membrane so actually water can go in and out, but not the solute, only the water. The water wants to be in a balanced environment with the solute. So where we have less solute, the water wants to go away from that side and go towards the side where there is more solute, because it wants to balance out the concentration of solutes. So that's kind of cool. So in some ways what this is the molecular pull of water. So it's like, this is when you cut a tomato and put salt on top of the tomato, and all of a sudden you got these water droplets on the tomato. The salt pulls the water out of the tomato. So salt and water are actually very connected. So water always follows the salt. But we can use this osmosis very nicely in many processes. For example, when we go into the tissue and we see that, look at the blood. The blood gets pumped by the heart, from the heart to the tissue. Once it's in the tissues, it diffuses into the tissues. And then how's it going to get back to the blood? How is it going to get back from the tissues into the bloodstream? The reason how it's doing that, it's not as efficient as a heart, but some bigger molecules, some solutes, are left in the blood. They don't leak out. And then those molecules, the albumin in particular, so albumin, they can drag the water back in at the end of the tissue where the fluid was dispersed in. And then it brings it into the venous system, and the veins bring it back to the heart. So that's one place where we use this osmosis very heavily. And it's a hard one to understand. If you don't get the osmosis concept quite well, mow it around it until Wednesday, then bring up some questions, or somebody might be able to explain it well. And then filtration, I don't know if you drink coffee. You drink coffee?
Oh, yeah.
Yeah, filtration. That's drip coffee. Yeah, if you drink espresso, it's a little different. But if you drink drip coffee, that's basically it. The water and its all particles are pushed through a membrane and a pore system, separating particles from solution. That's what that's like. You have that basically, for example, in the kidneys. In the kidneys, the interesting thing about the kidneys is basically a huge filter, about this much filter, that has very small little slits. Of course, it's all wrapped up in the kidney, but it's a large area of filter, and not much can go through. And so a lot of it is filtered out, the larger molecules. It's a size question. How big they are they're filtered out. And that's basically the same as the coffee filter. And then let's see what else we got there. And that brings me more to the active part. And we have, in the active parts, now we need ATP. So when we look at needing ATP, we're considering that we have to do some work to get this molecule from one place to the next place. So we don't use the concentration gradient to accomplish that. We're actually usually going against the concentration gradient. These are ways to establish concentration gradients. And so the pumps are really cool. They are active protein carriers that transport small molecules against the concentration gradient to establish the concentration gradient for later use. For the famous example is the sodium-potassium pump. And so here you've got a cell membrane, and you've got this thing, this molecule in it, that on one side can pick up 3 sodiums from the inside, and what it does, it throws them out to the outside of the cell. So it pumps them from the inside to the outside. And then it does the same thing with potassium, but it takes the potassium from the outside of the cell and drags it to the inside of the cell. So that's a pump, an ever-revolving door. It goes like this. Pick up the sodium, bring it to the outside of the cell, pick up the potassium, bring it to the inside of the cell.
So they just store it?
And so, huh?
So do they just store it then?
No, they don't store it. They continuously pump it.
Okay.
So they continuously ship 3 sodiums to the outside of the cell. This is a better example. So they continuously ship the blue is the sodium, ship the sodium out of the cell and make sure there's more on the outside than on the inside. And the same as the potassium will pull it to the inside of the cell. Potassium goes to the inside. And so there's more on the inside than on the outside. And then this is our perfect thing where we can establish this gradient that we talked about for ions. And since ions hold a charge, we can then use it in a nerve impulse, for example, say like, well, we need to bring in electricity to the system. Well, let's take these ions because they have a little charge on them, and that's a little bit of a current. And we can have all these sodiums, when we open the channel to let them in, when the nerve impulse happens, they can all come in now because they've been previously established that there's so many more on the outside. So the nerve impulse at the end of the day is a passive event. But the establishment of the gradient, so the concentration, so a lot of the sodium ions on the outside and very few on the inside, that is done by the pumps. That is an active process. But it would be really slow if you have an active process going on. It's like, oh, wait, the nerve impulse comes, we have to pump some sodiums in or some sodiums out. It wouldn't work. It would be way too slow and sluggish. So that, we'll talk more about that, I guess, in a few months in terms of the nerve impulse.
Where is that used?
That, everywhere.
Heart?
Everywhere. Because you got, these are sodium potassium is the big pump, but we also have a calcium pump, we have proton pumps with multiple pumps. And so we insert those wherever you use that, wherever you need that. So these pumps are kind of cool. Mostly we talk about the sodium potassium pump in here, because that's what is most needed to be discussed. And then when you look at larger molecules, you know, you can't just have glucose channel or a pump or a pore. You're talking like, well, you know, what are you going to do with viruses and stuff like that? I mean, large stuff. How are you going to bring that in? What we do is we have cell membrane that wraps around the stuff. So cell membrane becomes very important. When the Golgi sends stuff out, the Postal Service, the cell membrane fuses with the membrane that holds the stuff in that we want to export, fuses with the cell membrane and lets the material go. When we need to bring in things, it's the same thing. We wrap cell membrane around the substance that needs to come in, and we pinch it off, and then it goes to the inside of the cell. In a vacuum, protected. And then very often, we can fuse it with a lysosome and see what's on the inside, and dismantle what's inside. And then we can do with it what we need to do. But we have these terms. We have the term for cellular eating, and we also have the term for cellular drinking. What's the cellular eating term? Phagocytosis. So, bringing substances to the inside of the cell as a overarching term is endocytosis. Letting things go is exocytosis. Exo is like an exit. Endo always means one's own. That's why you know, bringing it in. Like the endorphin, for example, is an endogenous morphine. That's your own morphine. Yeah, just go for a run. You got a morphine, just go run. That's the same thing. But then when it comes to eating and drinking, you see the term cyto is all there with all of them. But phago means eating, and pinot means drinking. And I always remember that because Pinot Noir is a wine to drink. That's basically about it in terms of. Here I do write down that the lysosome comes into play, so you have to write it down too. Oh, that's the other thing.